Okay, so our next speaker is Ilya Nemenman. He's right now at the Emory University Department of Physics and Biology, where he is now a distinguished research professor and the director of the Theory and Modeling of Living Systems Initiative. He is an APS Fellow, a member of the Aspen Center for Physics and the James McDonnell Foundation Complex Systems Scholar Awardee. Before joining Emory, Ilya was he graduated from Princeton and then was trained in Kavli Institute for Theoretical Physics at UCSB and also at Columbia University Medical Center. And then he was a permanent staff member at Los Alamos National Lab. His work is on understanding how physical limitations on information processing shape biological organization, and work involves from paper and pencil theory to numerical simulation and analysis of experimental data sets. With that, I would pass it to Ilya.
Hi, can you see my slides and can you hear me?
Yes, we can.
Okay, perfect. Well, thank you very much. First of all, Yuhai and all the other organizers who invited me to give a talk here. And sort of without much ado, I will just jump into my talk. What I would like to ask you is that if there are any urgent questions, please ask them in Slack, and maybe one of the moderators can actually ask them in the middle of the talk, because it's sort of very difficult talking to an audience of people who are not from your field generally. So I don't know what it is that seems to be obvious and trivial to all of you sitting there, and what is not trivial at all, and I'm jumping over. And so if there is some clarification questions, not the large scale discussion questions, but some clarification questions, please ask them as I go, so this will save us some time. Okay. So first of all, what it is that my group does. We're interested in sort of two large-scale questions. First one is how biology processes information and learns. We are not wed to any specific systems in biology. We deal with neurons, with individual cells, with brains, with populations of animals. We try to understand what are the limits to the information processing capabilities, what are the computational and biophysical mechanisms that biology learns in order to uses in order to learn from its environments, right? So that's sort of one set of questions. The other set of questions is we are trying to understand how do we build models and theories of biological systems. And for this, the first thing we need to do is we need to understand what matters in a specific biological system for a specific question that you're trying to study. And so we need to understand, in all of those gory details that are, millions of proteins inside even a simplest cell, as non-detailed phenomenological descriptions of this biological phenomena that preserve the features that are relevant to whatever it is that we want to study. And then could we somehow use modern machine learning to actually extract those features and build the theories of the underlying systems automatically, right? So that's what we do, and I will give you a couple of examples from both of these pieces of work that we do. And so this is how the talk is going to be laid out. I'll first talk about why information theory, information quantities are useful for understanding both of the questions that I'm dealing with. And then I will try to very briefly, sort of without going into too many details, go into three specific applications that we have worked on. One is in figuring out what are the missing features in a certain cellular signaling system, then trying to figure out what are the relevant scales, what are the structures in the neural motor codes in vertebrate animals, and then trying to figure out how to use information in order to do renormalization group calculations directly from data. And so this is the three things that we will try to talk about. So first, why do we use information for what we study? And I want to focus on two properties of information that will be very useful for us later in the talk. First of all, information between two variables x and y is zero if and only if the variables are independent. Unlike correlation, which can be zero if the variables are dependent, like for example x and x squared, right? Information is zero only if the variables are totally independent. And the other beautiful property is the data processing inequality. In a Markov chain, if x, z, and y form a Markov chain, then the mutual information between x and y is smaller or equal to the minimum of the mutual information between X and Z and Y, right? These are obvious what properties. We know what they, we know them, everybody knows in this call what these properties are. And what I will try to argue is that they can be used not for the communication aspects of information theory, but rather they allow us to use information as a sort of statistics for understanding whether we know enough about the system to actually build a model for that system, right? So Shannon developed information theory as a communication theory. We are looking at it as a tool for detecting, do we know enough, okay? And so let's look at a couple of examples of how we would do this, a couple of sort of very abstract examples. So suppose I measure a large set of variables. For example, and this is X, right? This could be variables such as the speeds of all molecules of air inside the room. And what I want to do is I want to predict something relevant feature, something that I really want to predict. Let's say the pressure of the gas in this room. And so for that, what I would presumably want to do, I would like to try to compress the original data that I've collected, all of my X's, into some compressed variable Z, the things that actually matter, right? The summaries of the behavior of this molecule that matter for predicting the pressure. And this, in this particular physical case, this would probably be something like the temperature, the square of velocities of all of the individual molecules averaged, right? This is a dimensionality reduction problem. And most of the dimensionality reduction problems that we know of are linear dimensionality reduction problems, but in this example, it's not linear, right? We're interested in an X squared averaged. And so some, things like PCA and so on and so forth will simply not work, right? And so by dealing with information style quantities, what we can do is, at least in principle, and I will show you some examples of this in practice, try to solve a problem and understand what are the relevant features in this field, in the set X, that are useful for predicting Y, right? And the way that we would do it is we will look at multiple different possible compressions of X into Z, and then maximize the mutual information between Z and Y. And that is, we are optimizing the relevance, right? Very similar to what Tali Tishby has talked about. In fact, this is sort of the same bottleneck framework. We want to optimize the information between Z and Y, and we're guaranteed that we will not overestimate the relevance because of the data processing inequality. We cannot just by chance pick up something, say for statistical fluctuations that seems to work. And what we also know is that once we picked up the information between Z and Y, which approaches the information between X and Y, then we know that we have not only provided a good compression, but we've also provided a nearly perfect compression. We cannot do any better than recovering all of the information between X and Y in the variable Z, right? Another way of using information is a slightly different context, right? Suppose I measured something, variables X, right? And in this case, let's think of it as positions of planets moving around the sun. And we're trying to figure out what these planets are going to be doing a year into the future. That's the variable Y that we care about. Do we know enough to make that prediction? In fact, of course, we know that we don't know enough in this particular case. There is this X prime variables, which we just happened not to have measured, the velocities of the planets and the actual force fields, right? But how do we detect that, in fact, we do not know enough, that we have missed a bunch of important things in our data and potentially in our models, right? And again, we can try to use information style approaches as sort of a statistics for knowing that we have not measured enough. If I have figured out that there is some sort of intermediate variable Z that sits somewhere between X and Y, and I have calculated the information between X and Z, and this information turns out to be smaller, right, than the information that I need in order to predict the quantity of interest, the Y, right, then I know that I haven't measured everything. So the idea is that if I cannot predict acceleration from the position of planets, then presumably I haven't yet measured what I need, which would be in this case the forces, right? Or if there was friction, maybe also velocities, right? And so by looking at various intermediate quantities, which sort of form a Markov chain between what you've measured and what you want to predict, and seeing that you do not have enough information to even predict the intermediate quantity, then you know for a fact that that you're missing something relative to prediction of the thing that you actually care about, right? So that seems to be pretty obvious. So how do we actually use these ideas in specific context of trying to analyze biological systems? So now I'm going to move to one of the examples where we'll talk about missing features in cellular signaling. And this is, I'll motivate this work with, this is an example of a certain cellular signaling, protein signaling pathway called the nuclear factor kappa B signaling pathway. So roughly speaking, if you cut your finger and sort of things turn pink around the cut, this is inflammation. This is the activation of this nuclear factor kappa B. It's responsible for the inflammation. And then depending on the details, it's going to do a lot of different things. Like, for example, correct the regulation of the sort of activate the tissue remodeling, start cellular stress responses, issue cytokine storms, start removing free radicals, and all various different things, right? And so the idea here is that the mutual information between the behavior of what the cell is going to do, if you look at this list from what the cell could do, which I pulled out from Wikipedia, about 20 different things, it means that the input signals have to push through this pathway ballpark log of 20, let's say 4 bits of information, right? Can they really do this? Do we actually know enough? Is it just the nuclear factor kappa B? Is it just the tumor necrosis factor TNF signaling, or is something missing, right? We need to have about 4 bits pushed through. How many are there actually, right? And so we go and we pair with experimentalists, and this is a group of Andrei Levchenko at the time at Hopkins and now at Yale, and the graduate student Raymond Cheng. And what we do is they use optical imaging techniques to measure distributions of activation of the nuclear factor kappa B in response to the different concentrations of the tumor necrosis factor signaling, right? So what we're plotting here are the conditional distributions of the TNF, of the NF-kappa B response conditioned on different levels of the nuclear factor, of the TNF signal. And then we can calculate what is the channel capacity of this pathway. For that, we need, of course, to optimize over all possible distributions, input distributions of the signal of the TNF. And we can do that, and we have done this, and we get that the amount of information is about 0.92. I think I missed the 2 here, not 0.9, but 0.92 plus minus 0.01 bit, which means that it's much, much smaller than 4, which means that something is missing, right? That the simple picture of a linear pathway where a signal is a tumor necrosis factor, and then from there you go to nuclear factor kappa B, and from there you go to a whole set of all possible cellular responses. This picture is missing a substantial amount of information, about 3 bits out of 4, give or take, right? I'm not going to actually push on the actual number, but it's definitely missing something. And then we can start figuring out what it is that it could be missing. We can see maybe there are multiple different pathways that, starting from the TNF signal, actually lead to the cellular activation, not through just NF-kappa B, but through some other intermediate molecules. We do very similar analysis, and we still see that we're coming up short. Then we say maybe there is some negative feedback, and as always, very often happens, it doesn't help. Still we're coming up short. Maybe what the cell is doing is some kind of temporal integration of the signal, not just sort of one input, one output, but a time series of input and a time series of output. And you almost can make it work, almost transmit the right number of bits, but not quite. I mean, maybe you'll be able to do 2, but still not 3, 4. And so the only 2 things which are remaining are that either maybe you have what we call MIMO, like multiple inputs, multiple outputs, a very complicated network of interactions, multiple signals and multiple responses on the back end. And we're just looking at a single pathway while there is a whole network of interactions. That is possible. And we have only recently in the last few years with one of my former students, Vijay Singh, started trying to calculate channel capacities in various large, complicated multiple input, multiple output signaling networks. And the other possibility is maybe what's happening is that the cells are not independent, but many cells are measuring the same signal, the TNF signal, and each one of them has responses, and then somehow they talk to each other by secreting cytokines, and they come to a consensus of what it is that they're seeing, right? They are voting effectively. And so is it possible? And in fact, if you start calculating the information as a function of the number of cells that you allow to talk to each other, and granted, we are actually not sure if the cells do talk to each other. We just say, what if my response variable is not a single TNF, an NF-kappa-B concentration, but a vector of NF-kappa-B concentrations consisting of responses of 10 cells or 20 cells or 30 cells. Then after about 20, 30 cells talking to each other, you start seeing that the total number of bits that these cells can carry about the external signal starts approaching the number that we are looking for, which is, a couple of bits, 2 bits, 3 bits or something like that, right? And so in principle, it turns out that the only two options first of all, what we realize is that the information is missing, and there is two options that we know of how to recover the missing bits, what it is that we are missing in the model, which is either multiple input, multiple output responses among different cellular pathways inside the cell, or it's multiple input, multiple output responses among multiple cells, right? And so that's the idea that we had dealing with this particular system with the inflammation. And then we tried to figure out, can we actually show that the cells talk to each other when they're making the decisions? And it turns out it's very, very messy, not quite possible to do in this particular experimental setup. So, being theorists, we're kind of promiscuous in terms of which particular systems we work on. We choose the ones that are useful, right? Not the useful for the question, not the ones that are responsible for a certain disease. And so we switched to a different system to try to see if we can actually observe a collective communication, information processing in that system. And the system is development of mammary gland in the mouse, in a mammalian organism, right? And so here, what happens if this is the connective, sort of the schematics of the connective tissue of the mammary gland, and you have the beginning of the growth of the mammary ducts, and then they start branching, they start forming what's called these terminal end buds, this sort of small dots at the end. They grow into the tissue and form this basically space-filling network by about 10 weeks of development. And one can start asking questions, why do we actually need to have buds at the beginning? And it turns out that if you start measuring things, that these buds respond to gradients of the chemical signal, which is what tells, what polarizes this organ and tells it where the beginning and where the end is. So buds responds to gradients of this chemical signal, the epidermal growth factor, to which individual cells are not allowed to respond by fundamental physical laws, right? So if you think of a cell living in a gradient of epidermal growth factor, then the typical cell would have about 1,000 for physiological concentrations. It would have about ballpark 1,000 molecules of EGF to sense, and maybe the left side of the cell is going to sense 498, and the right side is going to sense 502. Right? This is real physiological numbers, but of course, everything comes with square root and fluctuation. So it's really 498 plus minus 25 and 502 plus minus 25. So you really cannot decide whether you should move left or right. So cells cannot do it, and yet buds can. And this is how these buds, terminal end buds, look like. They're about, ballpark 10 cells across, forming at the tip of the growing mammary duct. So what's going on there? And so we proposed a model which basically says that cells communicate with each other to make a decision where to go. And the idea of the model is rather simple. It's based on the previous theoretical work on what's called LEGI model, local excitation global inhibition model by Levchenko Iglesias. And for this work, again, Andrei Levchenko has done his lab done all of the experimental measurements. But the idea is that you have a concentration C outside of the organoid of the cell of the bud, and the receptors on cell surface, and each of these circles is a cell, and these receptors activate two types of molecules. One type of molecules is X, and this molecule X does not diffuse. If the molecule got produced in this cell, it stays in this cell. If it got produced in this cell, it stays in this cell. And there is another molecule Y, green one, which diffuses from the cell, across the cells, right? It goes through what's called gap junctions, really literal small holes that connect one cell to another. And so this way you are getting an average concentration of Y across all of the cells, right? These things, the spatial averaging, and then chemical pathways will compare the local concentration, the X, with the global concentrations Y. And if the local is larger than the global, then it means that you are on the head end of the gradient. And if the local is smaller than the global, it means that you are on the tail end of the gradient. And so you will not move in that direction. And so you can write down stochastic differential equations corresponding to this model. In the small noise approximation, this turns out to be actually a Gaussian relay channel, right? Because what happens is that the average concentration is measured by each one of the individual cells, and then it's communicated to the end cell over here, which is going to make a decision whether it's at the leading edge or at the trailing edge of the gradient, and make a decision which way to go. And so it's a very peculiar, nonetheless a Gaussian relay channel, peculiar because you are only allowed to do communications of a type which is diffusive, right? Which is physically constrained. You cannot have an arbitrary input-output mapping, but just a very specific, physically realistic communication between both the signal and the cells with each other. And for this channel, we can calculate the signal-to-noise ratio, and then we can use the signal-to-noise ratio to calculate the channel capacity. I'm not writing down all of these formulas. I had to show this one because it has sort of this logarithm inside. It's a logarithm of the number of effectively communicating cells, and every theorist knows that once you that getting logarithms in computations is in analytical calculations is rather hard, right? We usually do things in power expansions. So whenever you get a logarithm, a theorist inside you starts, clapping his hands, right? And so I had to show that. And so this is a first calculation, as far as I know, of a channel capacity for a biological multicellular information relay. And we can then ask, does it agree with experiments? And so we can actually develop models, right? Organoidal sort of small pieces of the terminal end buds, which have different orientations relative to the gradient, have different sizes, are in different points in concentration profile, and we can sort of compare. I'm not even going to go through this plot, but the point is that there are experimental data, and then the solid lines, which are predictions of our theory, and then there are dotted lines, which are predictions of theory that doesn't account for communication among cells, and it's clear which one agrees with data and which one does not, right? So it seems that communication matters here, and we can even estimate that the number of cells that can effectively communicate sort of what it is that I'm measuring here in this relay before the information gets degraded as in a standard sort of telephone game, is about 4 cells, right? So it's a 4-cell radius ball that is allowed to communicate with each other. And I remind you that the terminal end bud is about, give or take, 8 cells across, right? So it's about the same diameter as we predict the cells can actually talk to each other. And so this sort of ends the first part of my talk. We have started with this information theoretic analysis to identify that there are missing features in the data and the model of cellular communication, of cellular signaling. The missing features were cellular communication, and then we, in a different system, calculated the channel capacity of this multicellular communication system and showed that predictions agree with data. So that's sort of one way of using information as a useful statistics. Another example that we are going to look at right now is neural motor codes. And so the way that I'd like you to think about this is the following. This is a Rosetta Stone, right? And we need to decode it, just like if I'm looking at if I'm recording from a brain of a bird, as we will be doing in this particular example, there is a sequence of spikes that we're measuring, and we need to decode it, right? We need to understand which particular spikes correspond to which particular motion that the animal movement that the animal is going to do, right? It's a motor system. But before doing that, before doing the decoding, we need to first figure out which kind of language it is. In the Rosetta Stone, the biggest question that people had to fight with before decoding it was whether the language was hieroglyphic or whether the language was phonetic or ideographic, right? Which set of hieroglyphs, what do they correspond to a whole concept or to a letter or to a syllable or to words, or what do they correspond to? And in many systems in neuroscience, we're still at the stage where we don't know which kind of language we're dealing with. So this is, for example, recordings from a certain neuron inside the brain of this bird. Different times that this bird sings a certain syllable in its song. This is a songbird, Bengali finch, beautiful songs, right? So it sings a specific syllable, and this is every time it sings a specific syllable, you get this sort of bursts of activity. We can recode this activity into zeros and ones, which is if there was a spike or there wasn't a spike at a certain time point, and then somehow this activity codes for this particular song, right? This is the actual transcription and musical notes of the song that this bird is singing. And so what we need to understand, what are the features of this code that are meaningful? What are the timescales on which this code needs to be read, right? Is a certain spike important? Is a combination of spikes important? Or is it a number of spikes in a certain window, and how big of the window, right? This is all parallel to what I talked about in the context of Rosetta Stone. So how do we do this? So this is the work that we did with Sam Sober, one of my most active collaborators nowadays, and multiple students in his group. And so the idea is that we're trying to understand which features of the spike train are meaningful in encoding the behavior. And the behavior I'm talking about is this. So you hear, I hope that some of you have you heard the song? Was the song audible?
Yes, it was.
Okay, so it consists of the syllables, right? That's what I wanted you to see. And so what we need to understand is which features of the neural variation actually encodes variations in behavior, right? And you can sort of, there's two extremes. I don't subscribe to this trend, to this sort of this extreme, maximalistic point of view, but in the neuroscience of the late 20th century, there was sort of these two extremes, extreme ideas, which is that the code is the rate code or the timing code. And the idea would be that if I have two different types of syllables, maybe all of the neural sequences like 1, 2, 3, which have the same number of spikes, 3 spikes, right? But at different points in time correspond to one behavior, and all features 4, 5, 6, all spike trains 4, 5, and 6, which have ballpark 10 spikes, correspond to another behavior. And that would mean that it's the rate, the number of spikes that actually codes for the behavior. Another possibility could be that I always have 4 spikes in all of these combinations, but every time I have spikes in the pairs, of 2, 2, and 2, I would get a rendition of type 1, and every time I have spikes separated by larger distances, I will have renditions of type 2. And so that would mean that it's not the number of spikes that matters, but where exactly the spikes are that matters. And in the continuum between these two, what you can think about is what is the relevant time scale that matters, right? So on the left, it's the time scale of the entire spike duration, ballpark 40 milliseconds, right? We know here that it's a 40 millisecond window of activity in the brain that controls, let's say, the pitch of the subsequent syllable that the bird is going to sing. Or here, if we're counting spikes, like where exactly the spikes happen, exactly maybe down to a 0.1 millisecond accuracy. So it means that the temporal resolution down to a 0.1 millisecond matters, right? So anywhere from 40 to about 0.1, right? And so what are the relevant features, temporal features in the code? If I were to reduce the millions of neurons in the bird brain into some dimensionally reduced description predicting the behavior, at which scale should I reduce it? Okay, that's our question. So how do we do this? We're going to look at a very specific part of this problem. We're going to look at the pitch. This is the syllables. This is spectrograms of the syllables that the birds sing. We're going to look at the fundamental frequency here and here and here, and look at those fundamental frequencies, record their pitches, their frequencies, and you see that there's first of all a variability of about ballpark 10%. But we know that this variability is not sort of, oh, things happen, but it's actually actively introduced by the bird into the song for training. The bird does reinforcement learning. It tries to sing, a bit more here, a bit less here, frequency, and then chooses what it likes and then sticks with this. And it wants to get things just right, because whether it's going to mate or not, it depends on how accurately it sings, right? And so we're going to look at this variability and see how is this variability controlled by the neural behavior. We're going to record from a nucleus which is called RA. It's one synapse away from the muscles. It synapses directly onto the motor neurons, and it's crucial for singing. And so what we will do is basically the recordings from these neurons. Every dot is a spike. Every line, every row is a new rendition of the same syllable. This is a syllable, this is a spectrogram of the syllable above. We're going to look at the pitch at this particular point, about 40 milliseconds after the syllable has started. We're going to just make it very, very simple for us, because we don't have much data, and estimating information quantities from data is a hard problem. So we're going to simplify the problem and just say that there is not a continuous pitch, but rather just two pitches, 0 and 1, low and high. And then we're going to look at the spike window over here, 40 milliseconds of spike in activity, and we're going to ask how much information is there at different timescales between the spike train and this response variable 0, 1, the low pitch or the high pitch. And what we're going to do is follow many other papers in the field. We're going to look at the spike trains. So these spike trains are different, right? They may be 40 milliseconds long, but at discretization of 40 milliseconds, they're exactly the same, right? 4 spikes and 4 spikes. At discretization of 20 milliseconds, they're still exactly the same. At discretization of 5, they start to be different, to look different. And so we're going to discretize our spike trains at different resolution. And then we're going to calculate the mutual information between these words that we produce, right? Here the words are 4 or 2, 2 or 1, 1, 0, 0, 0, 1, 0, 1. And we're going to calculate the mutual information between these words and the binary word of behavior 0, 1 as a function of the discretization. It's a very hard problem computationally to do. We know how to solve it. Please believe me that we're not overestimating or underestimating the information. And this is what we get. So at 40 milliseconds, there is almost no information, and the maximum information is about 1 bit, right? It's exactly 1 bit, because it's either above or below the median value. And then as we start changing the discretization, we go down to about 0.2 of a bit, right? So you can predict, if you look at 1 millisecond resolution, you can predict which pitch the bird is going to sing right now from an activity of an individual neuron. You can predict this pitch with an accuracy of 0.2 bits out of 1 bit, right? So somebody a few talks ago was talking about that one neuron cannot matter in a large animal. I beg to differ, right? Individual neurons matter, and here is a proof. So, well, this is a correlative demonstration. We just showed that if I want to compress the neural activity from this neuron, then maybe there is a lot more information than below 1 millisecond. I don't know, I just don't have enough data to sample that, but clearly sticking to 20 milliseconds or 40 milliseconds, you're throwing the baby out with the bathwater. You're reducing your dimensionality of your input, but you're reducing it, throwing away what you care about as well. So in order to get away from this just correlative results, we need to actually show that this that it's not only that the neurons are correlated, neural activity at this resolution is correlated with the behavior, but we need to show that it causes the behavior, and that's hard, right? You clearly cannot do it with just analysis. So again, you start talking with your experimental collaborator, and you realize that that they can do it in a slightly different system, but not in this specific one, where you can actually stimulate neurons and check whether the temporal precision matters or not. And so here we're dealing with exactly the same bird, but what we're doing now is recording from a different part of the bird. We're recording from a muscle, from a junction between a motor neuron and the muscle. And this bird is anesthetized, and we're looking at how it breathes. And what you see is that this motor neuron spikes here and there, right? You see every time the bird breathes in, out, so out, in, out, in. This neuron is spiking, and slight deviations in how specifically this neuron spikes result in slight deviations of this black line of the actual pressure inside the air sac of the bird from the average one, the red one, okay? And so can we show first that there is still an information in the activity of this single neuron, that motor unit that you're recording from, and the pressure? And yes, we can. This is 8 individual birds, and for 7 out of these 8 individual birds, we show that the temporal precision, sort of the information in the temporal position of the spikes is comparable to, or even in some cases much larger than, the information in the rate with which the neuron spikes, right? So the temporal activity information from the temporal part of the code is red, and from the just counting the number of spikes is in blue. And then what we can do is, I'm skipping that slide, we can go and start stimulating the bird with what we identified computationally as some of the most common and most variable spike patterns that are produced in this particular motor system. And this is 3 spike combinations about 20 milliseconds apart from each other, where intermediate spike is at different positions away from the first spike. They all have the same rate, 3 spikes per 20 milliseconds, but the actual pressure that will be the pressure difference that you will produce in a real live bird by stimulating maybe not exactly one but other one neurons with the current is clearly very different if you have spikes that are, the gap is 6 and 14 versus 14 and 6, right? It's a different pressure traces that you will see in a real bird, right? So it's not an effect of a single cell, but close to a single cell, and clearly timing matters down to about 1 millisecond resolution or so, right? So we not only discovered this using information style analysis, but we have confirmed that the code, that the correct temporal scale for looking at the neural code in this system, in the bird breathing and probably singing, is at the resolution of about 1 millisecond or so, right? 1 to 2 milliseconds. As a side note, we can also decode this code and actually understand what the bird is trying to say to its own muscles, but that's a different talk. So now the last part, which is remaining, I have about 8 minutes to finish this. So I'll talk about using information approaches to do what statistical physicists like me, I was raised as a statistical physicist, know and love, which is renormalization group, right? So for those of you who are not from physics here, what is renormalization group? It's basically, it's a formal approach for analyzing how physical systems change depending on a scale from which you look at those systems. It's the only approach that we have in physics, sort of literally the only approach, classical approach that we have in physics, for detecting in a large statistical physics system which variables and which interactions matter for the physics on large observational scales, right? Spins may be interacting or on microscopic scales in many, many different ways, but not all details matter. We know that physicists love spherical horse and vacuum style models, and we love them because we can actually figure out which details matter and which details not. It's not that we think that nothing matters. We know that something does and something doesn't, and RG is what allows us to figure out what matters and what does not. One specific approach to RG is what's called Kadanov's block spin or real space RG. And so what it does, if you look at the interacting components, you start blocking them together in groups and sort of integrating out this microscopic degrees of freedom. You start from a single spin in this particular case. You have a block spin of 4 spins and then a block spin of 16 spins. And you integrate out microscopic details while preserving the property, preserving the free energy of the system, and therefore you're preserving all large-scale correlation functions in the system, right? The thing that you can actually measure experimentally. So you're sort of trying to throw away microscopic details while still keeping all of the observable correlation functions intact, right? And so what would happen if you do this to a standard sort of Ising spin, two-dimensional Ising spin? If you start coarse graining below or above critical temperature, at exactly critical temperature, if you are at exactly critical temperature, you're going to stay at it. If you are below, you're going to go to a system which freezes, either all spins point up or point down. If you're above, you get the salt and pepper sort of picture where every spin is uncorrelated from all the spins around it. And it's only in the middle where you have long range correlations which actually persist as you do the normalization. And by doing this, we actually figure out which structure of interactions, which features of interaction are important, and we also understand what are the variables that we should care about. And specifically, it turns out that to describe the system, it's the mean local magnetization that is important, right? The third order moments, let's say, of the spins are not important, but it's the mean magnetization and the variance of magnetization are the things that are important, and the rest we can throw out. But in order to do this, in order to apply traditional RG approaches, we need to know the Hamiltonian, right? How the things actually interact with each other. And we need to know who interacts with whom, the neighborhood structure. And so here's, for example, a picture from a recording, optical recording from a hippocampus of a mouse from David Tang's group. There's clearly no Hamiltonian. And even though some neurons are close to each other and some neurons are far away from each other, we don't know who interacts with whom. They may have axons that go, millimeters away, right? And so can we develop a randomization group approach that would allow us to figure out what are the relevant features, what are the relevant fields, the relevant variables to describe the dynamics of these neurons on large scales without having to build a model first, directly from data? So that's our question. And so this is what we're going to try to do. We'll use information twice here. We'll first say that information is going to be our measure of locality, right? So in an abstract case, I'm going to look at just binary spins for now. If there is no lattice, okay, and I were to try to do Kadanoff style approach and block spins together, which spins should I block? I don't know who the neighbors are, but maybe what I can do is I can pick out 2 spins that are the most correlated with each other or have the highest mutual information with each other. And if they are the highest, it's probably quite likely that they are also strongly interacting with each other. And so I am going to compress them into my new compressed variable z. So this is from my introduction, the variable x, and I'm going to compress them into a block spin z. And we need to preserve something. What are we going to preserve? Well, we want to integrate out the microscopic scale, which means the most correlated things. And we want to preserve information about other scales, or at least maybe about the next most microscopic scale, right? So maybe for each of the neurons spins that I'm going to integrate out, I'm going to find another one with which it's the most correlated. And then this one with which it's the most correlated. And these variables are going to be called my relevant variables, right? I'm going to integrate out the most strongly correlated features in the system while preserving the information that they have about, that we retain about the next strongest correlated part of the system, right? So we're going to compress X's into Z while keeping the information that Z has about the Y's intact. That's the algorithm. And this is, of course, the information bottleneck that Talitishby introduced above in a very specific context where the variables X and Y's and the Z's are all binary. We need to choose beta. Yuhai was asking questions about this, and it turns out that there is a very natural choice of beta here. If beta is too high, then you are starting to compress noise, and we don't want to do this. And so there's sort of the maximum value for beta that one is allowed to use given the amount of data of samples that you actually have from this system. And so basically, this is the approach that we're proposing. It's not the only possible approach, but there are also not the only one way of doing RG. There are many, many different ways of doing RG. That's ours. Maybe there are others. And we know how to choose beta. Again, it needs to be large enough so that we retain all possible information about Y while still not compressing the noise, not retaining the noise. And now what I can do, I can actually apply it for testing to the two-dimensional Ising system. And so I will simulate a two-dimensional Ising system that has locality and all, and I'm going to scramble all of the spins and pretend that I don't know that they come from two-dimensional Ising and run this data-driven RG on that particular system. And what we see is that we immediately recover that the system can exist in multiple different phases. If you calculate the correlation between these blocked variables, they either go to 1, which is, you are frozen, or to 0, which means that you are in a salt and pepper phase, or you stay more or less constant, which means that you are at a critical point. We can also...
Yeah, you have 1 more minute to wrap up.
We can also recover the actual scaling of the correlation functions in the systems, and we can recover that the order parameter in the system is the local monetization, even though we didn't even know that locality exists and that we should be doing some kind of averaging to get the average local monetization. And so this gives me my last recap, is that I can recast even RG in terms of using information quantities by defining a scale or locality using both the scale and the relevance using information, and we can recover properties of the two-dimensional Ising model from data without actually knowing the model. And applications to real data, real neural systems are forthcoming. And sort of my summary of the talk is what I showed you today is that information allows us to figure out if we missed some important features in our system. It allows us to figure out which features that we have measured actually are worth keeping in our description of the system. And maybe there are some hints that there is a formal data-driven information-based recasting of a normalization group, which will allow us to figure out what matters on large scales from these huge, large dimensional recordings that we now have in biological systems. And I'm stopping here. Thank you very much.
Thank you.